Good morning, ladies and gentlemen. It's my pleasure to welcome you to the Baker Institute and to Rice University, all you stalwart people who have come out so early this morning. First, I would like to thank Dr. Kirsten Matthews, the Baker Institute Fellow for Science and Technology Policy, and our colleagues at MD Anderson for organizing today's event. Science and health policy are major topics addressed by the Baker Institute through our Science and Technology program and our Health Policy program. The Science and Technology Policy program is run by Dr. Neil Lane, our Senior Baker Institute Fellow, who is President Clinton's Science and Technology Advisor and head of the NSF, and Dr. Kirsten Matthews. This program provides a space for policymakers and scientists to engage in a substantive dialogue with the hope that policy will more accurately reflect and be more consistent with current scientific knowledge. Over the past 7 years, they've engaged in discussions on topics such as improving international scientific collaborations, funding of scientific research and development, and stem cell policy. The program is known for bringing leaders in science and public policy to Houston to engage with Rice students, the Rice faculty, as well as local business and community leaders. The Health Policy Forum, run by Baker Institute Fellow Dr. Vivian Ho, who's also with us today, studies the ways in which economic incentives and government policies influence the quality and costs of health care, obviously a primary issue in today's national debate. Today's program is part of the Medicine Research and Society series co-organized by the Baker Institute Science and Technology Policy Program with the University of Texas MD Anderson Cancer Center. The Medicine Research and Society series is dedicated to educating the public on the important intersection of science, medicine, and policy at the state and national levels. Co-sponsors for the event include Rice University's Institute for Bioscience and Bioengineering, as well as the Baker Institute's Health Policy Program. This morning's event, Personalized Medicine, Genetics, and Healthcare, addresses the impact of the Human Genome Project and genetic research on healthcare, and we are honored to have our distinguished speakers share their perspectives with us on this subject. Over 10 years ago, the first draft of the Human Genome was released after many years of extremely hard work, under budget and ahead of schedule, just like the federal government's budget. Now scientists are aiming to utilize this information to predict disease and determine how people will tolerate drug therapies. But with the enormous investment in this project, approximately $3 billion, some question if the expense was worth it. Even Craig Venter, a genome sequencing pioneer, has said that, quote, "We have in truth learned nothing from the human genome other than probabilities. How does a 1 or 3% increased risk for something translate into the clinic? It is useless information." That's quite a contentious remark. So our speakers this morning will help us understand the human genome, what information was obtained from the Human Genome Project, what researchers are currently working on, and what the impacts are of genomic research on healthcare now and in the future. Now, I would like to introduce my friend, Dr. John Mendelsohn, President of the University of Texas MD Anderson Cancer Center, who will introduce our speakers. Under Dr. Mendelsohn's outstanding direction, MD Anderson has taken a leadership role in translational and clinical cancer research and has been named the top cancer hospital in the United States for 7 of the past 9 years. John and MD Anderson have been great collaborators with the Baker Institute over the years, and we're honored to work with him and his colleagues. Join me in welcoming John to the podium.
Well, thank you, Ed, and let me begin by thanking Ed Jerigian for bringing symposia and meetings like this for all of us in Houston to enjoy and learn from. So you've added tremendously to our community, Ed. I also want to add my thanks to those who organized this meeting and mention Mark Marino and Ray Du Bois from MD Anderson, as well as Kirsten Matthews and Neil Lane from Rice. It's a wonderful collaboration. So why are we here today to talk about personalized genomic medicine, genetics in healthcare? Very briefly, there's been a confluence of three major research areas in the past few years that have just changed what I think is happening in medicine today. First of all, and I'm looking at it through the lens of cancer, we now know what causes cancer. It's caused by the abnormal function of the genes that control cell proliferation. And the number of genes that control cell proliferation is probably a few hundred out of the 23,000 genes in the genome. Remember what the genome is. It's all the genes in one person's genetic information base. So we know, we've identified, we can name the genes that cause cancer. That's the first thing. The second thing is that technology and instrumentation has increased so incredibly during the past 20 years that, for instance, the Human Genome Project, sequencing one genome, took 9 years and cost $3 billion. Today, we can sequence one genome in 3 weeks for $10,000, and it's estimated that in 2 or 3 years, we'll be able to sequence one genome in 1 week for $1,000, and that's half the cost of an MRI scan. So this has come to the point where in real time and with technology that is accurate, we can sequence genomes. And that allows us, for example, to look at what have we inherited in our genes that increase risk of disease. And that's what Dr. Venter was referring to, because in most cases, the genes we've discovered only increase risk slightly, although some genes, if you inherit them, increase risk a great deal. But the third thing that's happened is the drug companies and academia have begun to target that list of a few hundred genes that cause cancer. Today, there are 800 drugs in the pipeline that are designed to attack the products of these genes that can cause cancer. 800. And that means that we are now able, for the first time, to take a patient's individual tumor and interrogate it and find out which genes are abnormal in that patient's tumor from that list of 200 or 300, and then select drugs that were designed to target the products of the genes that are abnormal in that patient's tumor. And the response rate isn't 2 or 3%, it's 50% or higher. So Dr. Kurzrock, I'm sure, will get into that in detail. So that's why I'm excited about the opportunity today, the fact that all of the science we've funded in this country and all the hard work the scientists have done are leading to something that I think will change medicine. But let's hear what our speakers have to say about it, and then we'll have questions that you can ask. Our first speaker is Eric Green. He's the director of the National Human Genome Research Institute. That's the institute that did the Human Genome Project, and that's at the National Institutes of Health, and he's been in that job for over a year. He received his MD and PhD at Washington University and had training in clinical pathology and laboratory medicine to prepare for what he's done. And he was a member of the Human Genome Center at Washington University, one of the 3 academic centers that participated in the Human Genome Project. And then he was recruited to the NIH, and for a while was chief of their technology branch and all of the intramural sequencing at the NIH, where he has had a major role in large-scale DNA sequencing to address important problems in genomics, genetics, and biomedicine. So we're all looking forward to hearing from Eric. Please come up.
Well, thank you, John. It's a pleasure to be here, and I'm going to extend this enthusiasm that you heard in some of the introductory remarks and tell you about really the course for what I describe as genomic medicine. Now, the truth is it's a very opportune time to be discussing genomics. It's been 20 years and 2 months ago the Human Genome Project began. And then 10 years ago, as you heard, and it'll be the 10th anniversary of this publication, it'll be February of next year, was a description of the first draft of the human genome sequence. And in fact, this notion that we are celebrating the first decade of having in hand a sequence of the human genome has been widely written about in both the popular and the scientific press. And the reason this is capturing so much enthusiasm, in part, is the recognition of the accomplishment of having sequenced the human genome, but increasingly it's the recognition that there's incredible opportunities for applying that knowledge to human health. And in particular, the notion of marrying genomics and medicine is not new. In fact, that was a fundamental promise of why we got funding for the International Human Genome Project. And both the popular press and the scientific press, even during the Genome Project, began to marry the idea of genomics and medicine in coming up with phrases such as genomic medicine. Now, by genomic medicine, what I mean by this is healthcare tailored to the individual based on their own unique genetic makeup, their own unique genetic blueprint, if you will. And this is largely synonymous with personalized medicine, individualized medicine. I've even referred to as precision medicine. Now, in thinking about this, which I do a lot, in charting a course for genomic medicine, being now the director for a little over a year of the organization in the world that is dedicated to the funding of genomics research, I've thought a lot about how we need to make a course successful from the fruits of the Human Genome Project to the actual realization of genomic medicine. Now, I'll be the first to admit in trying to programmatically think about the many, many steps that will be involved to truly realizing genomic medicine, we can't believe we know all the steps right now, and I'm sure there'll be some surprises. What I do know is that we were successful at the Human Genome Project. I got involved in it on day one and saw it to the very end. And now I'm passionately trying to help chart and navigate a course that will result in the realization of genomic medicine. And the major reason for doing, if nothing else, it will fulfill the promise of why we sequence the human genome in the first place. I'd make the very bold claim, I can do it on behalf of the Institute, that the central mission of NHGRI, the National Human Genome Research Institute, and I'd like to believe the field of genomics, really needs to be about establishing that path to the realization of genomic medicine. And so what I want to do for the next 10 or 20 minutes or so is to describe for you this journey, what has been accomplished in the last 7 or 8 years since the end of the Human Genome Project, and what we might be thinking about in the future. And in each of these steps, what you're going to see is that you successively take steps that get you a little bit more data, a little bit more knowledge, and with it comes a little bit more insights that inch you closer and closer to an era of genomic medicine. But I'm just going to have time to highlight a few of these. One of the very first steps that had to be dealt with was understanding how the human genome actually works. I'm not going to describe this in any detail, but the Genome Project delivered 3 billion letters of information, the order of the G's, A's, T's, and C's that constitute a hypothetical reference human genome. It said nothing about how to actually read those sequences and understand the language, the grammar, the syntax of how it all works. It didn't give us a list of the 20,000 or so genes that were within the human genome. But in fact, over the last 8 years, there have been tremendous advances in our understanding of how to actually understand how the human genome works. And importantly, through a series of international efforts, all of this information is being put out on the internet, and increasingly you can go to websites and not only get to the sequence of the human genome, you can get to detailed interpretations of that sequence. We'll be studying and trying to understand the human genome sequence for decades to come, like a great novel. But indeed, what we have accomplished in the last 8 years, and I think what we'll accomplish in the next 10, will get us far down this path of understanding how our genome works. But in fact, the next step that we've been pursuing is not understanding how a hypothetical genome works. We're interested in understanding how each of our genomes uniquely work. Each of us are roughly 99.9% identical at the sequence level, but what that leaves are about 3 to 5 million base differences between any two of us. The great majority of those differences are totally innocent, have really no consequence for us with respect to disease or health, but a small subset of those actually are quite relevant. And what we attempted to do over the last 7 years is to develop rich catalogs of these very common variants that exist across different human populations, have catalogs of those, and then systematically begin to study those and to see what consequences they might have. And the reason we want to study those variants is that very small percent of the nucleotide differences between our genomes actually are very relevant for human health, drug response, and disease. And there's been an impressive amount of acceleration through a whole series of studies I don't have to describe that have really advanced our understanding of human disease and the genetic basis for it. You heard in the introductory comments some of this has been particularly targeted at cancer, but in fact it goes the full gambit of diseases. Now, in thinking about diseases, and by the way, virtually every disease you can name has a genetic component, some to a greater degree than others. You can broadly divide them into two categories. There are what are known as rare genetic diseases, or these are simple because they're basically caused by a single gene defect, a defect in a single gene, as shown on this pie chart. These are diseases such as sickle cell disease and cystic fibrosis and Huntington's disease. And what I can tell you is that there are thought upwards of 4,000 or so such rare genetic diseases. And since the beginning of the Genome Project, we've now figured out the genetic basis for maybe about half of those. There's still another half to be conquered, and major acceleration is taking place there as well. But what we have seen in genomics is almost week after week, another rare genetic disease, the molecular basis of which is better defined. What was unclear was whether these diseases, which are more common diseases, and they're genetically complex because they involve not a single genetic difference, but multiple small genetic differences that conspire together with often as a greater contribution of the environment to confer risk for getting the disease. These are diseases such as hypertension, diabetes, cardiovascular disease, mental illness, asthma. In fact, it's the diseases that fill hospitals and clinics around the world. And yet, because of the genetic complexity of not just looking for one gene defect, but multiple subtle gene defects, scientists for many years wondered, would we ever be able to crack that open and really get at the molecular basis of complex genetic diseases? And while I can tell you the last 8 years hasn't brought complete insight into complex genetic diseases, advances have been made where we now have thousands of regions of the human genome that we now have narrowed down to very small intervals that we know contain variants that confer risk for all the diseases I just alluded to. And over the next 10 years, we firmly believe we're going to figure out what the causative nucleotide changes are in our genomes that give us that risk for that disease. And we believe this is going to greatly accelerate a tremendous amount of energies and accomplishments in the study of complex genetic diseases. So there's a lot that has been accomplished, and I'm happy to answer questions about it, but I don't have time to drill too deeply in it. One of the things that's going to greatly accelerate dissecting these complex genetic diseases that are so important with respect to worldwide health burdens is going to be the fact that we now have technologies that have really just come to pass in the last 5 years that are greatly accelerating our ability to sequence whole genomes and get complete inventories of all variants of individuals, eventually individual patients, but even during research studies, individual subjects, so we can figure out which of those genetic changes are the ones conferring risk for the kinds of diseases that we want to study. And in fact, this was all predicted to hopefully work as part of a major initiative that took place when the Genome Project ended. The quote earlier was that it cost $3 billion to sequence the first human genome. In fact, the actual sequencing of it only cost about $1 billion, $3 billion for the Human Genome Project, but the actual sequencing was about $1 billion. It sounds like a lot of money. It was the best money ever spent by the international scientific community. But we recognize that when the Genome Project ended, we couldn't just stop having sequenced one genome. The power of this would come from having technologies that allowed us to interrogate many genomes, eventually, hopefully, as part of clinical care. And through a program that was designed to accelerate technology development both in the public sector and the private sector, the goal was to be able to develop a technology that could sequence a genome for $1,000. And this became a battle cry for the community called the $1,000 Genome. Could there be new technologies that would eventually deliver the ability to routinely sequence a human genome for $1,000? I have been involved in genomics for 22 years. I have been overwhelmingly impressed with technology advances that have taken place in the field. I have been blown away by what has happened in the last 5 years in the arena of DNA sequencing technologies. There's not just one new technology or 2 or 3 or 4 or 5. In fact, there's about 9 different technologies you can buy now, each that are slightly better than the other, with probably another 4, 5, 6, 7 that will come available over the next 5 years. This is not an area where I'm at all worried about anymore. We have seen a massive improvement in the ability to sequence DNA, and it has resulted in a significant decrease in the cost of DNA. Let me show you some data. We actually, at NHGRI, our institute funds the 3 largest sequencing centers here in the United States, one of them right here at Baylor College of Medicine, and we have them report to us every 3 months how they're doing in terms of the cost of DNA sequencing. So if you just simply ask the question, what is the cost per generating 1 million bases of raw DNA sequence, and how has that been taking place over the last 10 or so years, we actually keep track of that data. So let me show you what that data really looks like. Notice, first of all, the y-axis is on a logarithmic scale, and notice the green line. The green line refers to something known as Moore's law. Moore's law is the law of the computer industry that says that everything in the computer industry gets better by a factor of 2 every 24 months. That's the Moore's law in green. Anybody who knows much about technology development will say, if you're keeping up with Moore's law, you're rocking. You're just doing fantastic. Look what's happened in DNA sequencing. Going back to 2001, our 3 largest sequencing centers for a long time kept up with Moore's law. That's the red graph, the first half. But then look how it just sort of drops off like the side of a mountain. That's when they implemented these new sequencing technologies. We now have blown Moore's law out of the water, and it continues up into the present time. And the fact of the matter is, in our journey getting down to a $1,000 genome, we're about here right now. It now costs something on the order of $10,000, $20,000 to sequence a human genome. And in fact, just by momentum, I predict over the next 3 to 5 years, we will get to the point of being able to sequence a human genome for $1,000. Now here, I will admit my optimism will slow down a little bit. Because the fact of the matter is the next step in our journey towards genomic medicine will involve being able to routinely analyze all the data that can now be generated with these new technologies. And the fact of the matter is, this is actually the reality of what we face. And what I think it is the current largest bottleneck in genomics. And that bottleneck is simply a reflection that these new sequencing platforms spew out so much sequence data so fast that we actually, as scientists and clinicians, can't even get our hands, our heads, our mouths, our hands, anything around it quick enough. It is really leading to what I know is the largest bottleneck in genomics right now, and that is the computational bottleneck. The fact of the matter is we can generate data in a fashion that just simply overwhelms the system. We don't, unfortunately, have enough infrastructure for actually handling all of that data and storing all that data. Even being able to push that data across the internet fast enough becomes a problem. We don't have enough software tools at the moment for being able to deal with all the data and all of its nuances. And quite frankly, and I hope people at Rice University hear this and other universities, is that we don't have enough people who are trained in statistics and computational sciences as well as biomedicine. And we need a larger workforce to help us in this area in the future. And so the fact of the matter is we have these incredible technologies, but even when we solve the issue of being able to handle all of the data, we will then face an informational bottleneck, one that I think is going to get a lot of attention in the next few years. Because these fancy methods that allow us to sequence DNA will spew out sequences of individuals, research subjects, and eventually actual patients. And with that will come lists and lists of the variants. Each of us carry about 3 to 5 million of these variants. Unfortunately, we don't yet know which of those variants are actually relevant clinically, and we will stare at them and we will ponder this for a long time. But in fact, if we actually go to deploy this right now in a real clinical situation, we really face a situation something like this, of having this massive sequence on these clinical subjects or patients, and not yet quite being certain exactly how to deal with it. This now represents a key area of what we need to be doing over the next 5 and 10 years. Harold Varma's previous director of NIH, now head of the National Cancer Institute, wrote a very nice piece commemorating the 10th anniversary of the sequence of the human genome in New England Journal. And I thought his quote was spot on. He said, "Physicians are still a long way from submitting their patients' full genomes for sequencing, not because the price is high, but because the data are difficult to interpret." So we're at that awkward stage right now where the technology has surged ahead of our ability to handle the data and eventually interpret the data. But I think all this will be solved. It's just going to represent what we'll be working very hard on in the next decade. But there's other steps. I haven't taken you all the way across this journey. There's, of course, issues of developing new diagnostics. Of course, we want to develop new therapeutics and new preventative measures. And as we inch closer, there's probably even other questions and other steps that we haven't quite faced up to. And many of you are probably thinking of those, and I'm sure they're actually quite real. The truth of the matter is this is a very interesting time for genomics, I can tell you. As somebody who's participated in genomics for 22 years, so much of what we've done has been about technology, has been about basic science pursuits, about computational biology, and so forth. And the marrying of imagery between basic genomics and clinical medicine is actually very new for many of us in the field, and certainly new to many of the scientists who have been deeply engaged, especially in technology development. There's many new challenges that come out, and we're going to hear about that from some of the other speakers, and much of this is new for us, but it's actually something we're very excited about. It actually is a good time for us because as an institute, we're right in the midst of a strategic planning process. We're actually near the very end of it. It's a time where we actually have really people look at us under a microscope and see what we're doing as a field and as an institute. We also spend a lot of time looking in the mirror, figuring out what are the best opportunities that we should be pursuing. I can tell you that strategic planning is sort of a part of the culture of genomics. When the Genome Project began, I can tell you I was there, we didn't know what we were doing. We had a goal, and we had no idea how we were going to get to that goal, sort of reminiscent of where we are now to some extent. And 3 published strategic plans guided us through the Genome Project from beginning to end. And then when the Genome Project ended, our institute put out a new strategic plan that has guided us for the past 7 or 8 years quite well. But it was time for a new strategic plan, and so the Institute has spent the last 2 years inviting literally hundreds of people in the community and even outside the community to come and talk to us, and we've enlisted our advisors, and we've spent a lot of time debating internally. And I could tell you we recognize that it was a historic moment. Will be coming up in February of next year with the 10th anniversary of this publication. And so that's when we've decided we're going to publish our new strategic plan, which you'll all be able to read about in February of next year. So I want to just spend the last 3 or 4 minutes just telling you a few highlights of what you're going to read about if you read our strategic plan in February. First thing I will tell you is that genomics is not just about the National Human Genome Research Institute. In fact, it's not, there's many other institutes at NIH that now pursue significant amounts of genomics research. There's other funding agencies in the United States that pursue genomics research. And in fact, there's many other groups around the world that are pursuing genomics research. So genomics and the strategic plan that we lay out is not just about our institute. It's actually about the whole world community of genomics, and it's even much bigger than what any one funding agency can contemplate tackling. One thing that was very interesting in talking to members of the community and getting lots of feedback from people was that we, for a long time, and especially when the Genome Project ended, we described the progression to genomic medicine as we would sequence the genome, we'd understand how the genome works, and then we'd apply it to human health and to healthcare. And what we got back quite strongly was what we needed a far more sophisticated way to describe that progression. That it was naive to believe that just once you understood the genome, you were then ready to apply it to human health and disease. And that discussion has really led us to describe in a much more sophisticated way what we think this progression is going to look like. And I just want to share it with you because we've now divided this progression into 5 major domains that get us sequentially from the base pairs of the genome to the bedside of patients. The first domain of research activities is understanding the structure of genomes. How is it that genomes are put together? We also need to do research to understand the biology of genomes, how they work. And as we have a better understanding about how genomes work, we could then turn our attention to using genomic approaches to understand the biology of disease. With new disease insights, one could imagine doing research to advance the science of medicine. But as all of us realize, just you have a great idea on how to pursue medical science, it doesn't always mean that when you put it into the ecosystem of healthcare delivery, it actually proves to be effective. So we also must pursue research that will prove to us that we can improve the effectiveness of healthcare. And each of these 5 domains really represent research activities that we need to be thinking about over the next 10 and 20 years. Now, I would be the first to admit it's not all about these 5 domains. In fact, there are what we call cross-cutting elements that are critically important and always have been important in genomics. For example, bioinformatics and computational biology, I already told you, is hugely important, a huge bottleneck, pervasive across all these domains. There are major education and training issues that go across all of these domains. Applying genomics all the way to the bedside of patients will have some important consequences with respect to how people are educated and how healthcare professionals are educated. And of course, there's many societal issues. I think our next speaker is going to touch on some of these, and there's many more that many of us can be talking about as we infuse genomics more and more into society. But what's been very useful in thinking about this construct of these 5 domains is to think about what have we accomplished the last 20 years in genomics, and what are we likely going to accomplish in the next 20 years. And so I just want to just share with you what ends up being the organizing figure of our new strategic plan, because I think it nicely captures a little bit of historic context and a little bit of prognostication of what to expect. And one way we found useful to represent it is to simply plot, hypothetically, the genomic accomplishments that have and will take place. And you could do this in the form of what's called a density plot. So it's basically those who don't look at these kinds of data. This is just a hypothetical plot with each blue dot representing a hypothetical genomic accomplishment. And as the dots pile up on each other in the graph, they successively turn green and then yellow and then red. So back during the Human Genome Project, where were our research accomplishments taking place? The first domain. We were understanding the structure of genomes by studying the human genome. And yeah, we learned a little bit about how the genome works, but just barely started to crack that surface. What about the last 7 or 8 years since the Genome Project ended? Well, we've continued to explore to help our understand the structure of genomes. But in particular, we've really started to open up our understanding of the biology of genomes through many research accomplishments. And we've even taken a few of those accomplishments a little bit further to understand the biology of disease. And maybe there were even a few home runs where these became clinically applicable. But those are very few and far between. What many of you are thinking about, what about the next 10 years? What's this going to look like over the next 10 years? Our prediction is that it'll look something like this. We'll continue to explore genomes, and we will certainly continue to now emphasize how is it that the human genome works and really understand the biology of genomes. We think accomplishments in the second domain are going to be even more spectacular the next decade they were in the last decade. But the other thing that will be immensely exciting about this next decade will be our ability to better understand the biology of disease. We think the center of gravity of genomic accomplishments are going to be in domains 2 and 3. We believe there's going to be tremendous amount learned about the molecular basis of human disease over the next 10 years. Some of that will yield productivity in the last 2 domains. Yes, we will advance the science of medicine, and there'll probably even be some more home runs where these are proved to be effective in healthcare. But in terms of realistically imagining changing the face of medicine in the next 10 years, unlikely. And just managing those expectations are important, because if you think about medical discoveries and then when they actually get to be applied clinically, often it's many decades before they're fully implemented and proved to be effective. We have great optimism we will get there. And beyond 2020, I think the center of gravity will continue to shift rightward and be increasingly finding its way into the 4th and 5th domain. And I think that's in particular where we could expect there to be some terrific accomplishments. But realistically, we're in this for the long haul. This is going to be a long, hard road to follow. But I have great optimism that genomics is going to find its way to the bedside of patients. So that's what I want to tell you. I want to leave you with a quote. Jeffrey Carr was an author who wrote a 5-part series in The Economist commemorating the 10th anniversary of the Human Genome Sequence. He interviewed lots of people in it. If you're actually interested in reading this, it's free. If you Google Jeffrey Carr, an economist, you can get to this 5-part series. He said a lot of really nice things, but I really like this quote. He said, "The race was to sequence the human genome, all 3 billion genetic letters of it. A race not to the finish, but to the starting line. The end of the Genome Project was the starting line, not the end point. But moreover, the new race marked by that starting line was a marathon." And I really believe this is an exciting and compelling marathon. I really do believe it's going to change the practice of medicine. But we need to be realistic. We're in this for a very long run. I hope many of you come along. We need a lot of thoughtful people involved in that marathon. But I think it's an exciting marathon nonetheless. So I will stop there, and I'm happy to take any questions now. Or are we doing questions later? Afterwards. Great. Thank you.
Well, thank you, Eric, for a marvelous, very accelerated tour through what's been happening. Our next speaker at this symposium is Gail Javid. She's counsel in the law firm of Sidley Austin, LLP, in their food and drug regulation practice that they carry out. She holds a JD from Harvard Law School and a Master of Public Health from Johns Hopkins University. So she's doubly looking at the problems that we're talking about today. In the past, she's been affiliated with centers of excellence both at Johns Hopkins and at Georgetown University, and is specialized in FDA regulatory issues and in the intersection between law, science, and policy, including direct-to-consumer marketing of genetic testing and FDA regulation of biotechnology. Gail, please come up.
Well, thank you, and thank you for inviting me to speak today. A little public service announcement. The views I'm expressing here are my own, not those of my law firm or any of the clients it represents. Okay. The arrow that points right. Oh, that arrow. Sorry. So Dr. Green gave us a great view of the universe, the universal map of the genome and the research agenda. I'm really focusing on a zip code, as it were, although it's an important zip code, almost as important as the zip code we're sitting in. And that's the zip code being FDA. The Food and Drug Administration has a mission to protect and promote public health by ensuring the availability of drugs and medical devices that are safe and effective. So it's a dual mission: to protect by preventing stuff that's not safe from getting on the market, and to promote by ensuring that stuff that's safe gets on the market. FDA has viewed pharmacogenetics, personalized medicine, and I'm using the term pharmacogenetics to mean the use of genetic information to inform drug development, selection, and dosing as a means to improve drug safety and effectiveness. So there's lots of interest at FDA, but a little bit of uncertainty about how to get there. And I think this is represented in a comment by the FDA commissioner saying, the old paradigms won't work. We're still trying to figure out what will work to get personalized medicine into drug and device development. So a couple of challenges that I'd like to talk about that are facing FDA and that have yet to be resolved. Diagnostics. If you want to have a drug that is informed by genetics, you need a test. So validated genetic tests are a foundational requirement for personalized medicine. But in fact, there are very few tests that have been reviewed by FDA, and even fewer still for personalized medicine. That doesn't mean the tests aren't out there, they're just FDA hasn't looked at them. FDA's stated ideal is a co-developed drug and test, but that ideal has not really been realized in practice, except in very few instances. You're all familiar with the HER2 test for Herceptin and EGFR for Vectibix and Erbitux. There's an unclear regulatory pathway for these tests, and the difficulty of coordinating the device and drug industries is also a challenge. Also, what do you do if the genetic information is discovered after the drug is on the market, which is the norm? FDA is very concerned about the use of retrospective data to change labeling. So going to the diagnostics development issue first, there's two paths to get a test on the market. There's the laboratory developed test, which is the norm, and the FDA regulated test, which is the exception. Test kits are regulated by FDA. LDTs, laboratory developed tests, are developed by laboratories. FDA has said recently, we are going to regulate what's going on in the laboratory, but they haven't really explained how they're going to do that. And it's very unclear what they're going to do and to whom, and the laboratories are really, really nervous about this, and there's been a lot of consternation, kerfuffle, and we haven't seen the end of this debate. It's been, you know, as long as I've been following this issue, it's been going on, and we're not exactly close to a resolution. But FDA at the same time is very concerned about the validity of genetic tests, particularly when they're informing drug dosing and selection. And they are signaling that drug companies developing tests will be required to ensure that there is a validated diagnostic if they want to talk about the genetics in the labeling of the drug. And if the genetic information is necessary to properly prescribing the drug, so it has to be in the labeling. That means in practice, if there's not a test, there's not going to be a drug. And so this could result in the delay of drug development. And in fact, there's a very good example showing that it is. And that's the Omepro drug, which I'm sure many of you are very familiar with. The company wanted an indication to treat CML in adults with a particular mutation who had failed prior therapy with Gleevec. The Oncology Drug Advisory Committee within FDA said, well, if we're going to approve this drug with that indication, we need a well-characterized assay to detect that mutation before we're going to sign off on the drug. And by well-characterized, they meant FDA reviewed. And the rationale was a well-defined companion diagnostic test available at the time of approval of any drug that claims to identify a subset of patients that will have a differential response to therapy is an integral part of the development of any drug that makes such claims. So FDA basically told the manufacturer, get us a test before we'll sign off on your drug. So the company is working on that. Other challenges, as I alluded to, it's more the norm than the exception that after a drug is on the market, more understanding of the genetic basis of response are identified. Well, so what do you do? How do you get that information into drug labeling? The ideal would be at the time of drug development, you save everybody's samples and then you can go back and look. And but that, so far, although I think companies are moving towards that, that hasn't been the way it has been. And FDA has expressed concern about looking retrospectively at data. So an example of this is the KRAS story, which again, I'm sure many of you are familiar with, where at the time of approval of these drugs, the labeling said you need to test for EGFR before prescribing the drug. And later it was determined there was another mutation at stake, KRAS. And so the sponsor asked FDA, can we relabel to talk about this? FDA, during some meetings, were reluctant to do so for a few reasons. These, again, was based on retrospective data. Not all the samples had been maintained, so only part of the subject population could be looked at. And the primary endpoint hadn't been met in the tests where they were looking retrospectively. So all this boils down to FDA concern about bias in reviewing the data under these circumstances. But at the same time, the practice of medicine kept moving forward, and ASCO recommended that this testing be done in the interest of patients. So FDA did ultimately approved this labeling change, and specifically to say that individuals with colon cancer who had these particular mutations won't respond to therapy with these drugs. And FDA didn't really explain why it changed its mind. In a speech, one official said, "We did so because the data plausibly showed that these drugs were not efficacious in patients with these mutations." And the companies were able to replicate that finding. But they did that, but there's a but. And I guess stepping back a second, why does it matter how the labeling is changed? Some might argue that doctors aren't really looking at the labeling, they're looking at the literature. So why does it matter? Well, there's a good debate there. But there's one very particular reason for the manufacturers why it matters, because you cannot promote in a manner inconsistent with the label. And I think this next example shows us a very practical reason why manufacturers care so much. So the label was updated to say that the drug is not recommended if you have a KRAS mutation. But subsequently, FDA took issue with the way a particular representative was conveying that in promoting the drug, saying KRAS status or lack of KRAS status is a positive predictor of clinical response to Vectibus. Companies want to say not only it won't work in you, but if you're not in that subset of people who have the mutation, it will work great in you. So it's an efficacy claim versus a safety claim. And FDA is even more skeptical about efficacy claims than safety claims. So I know it sounds a little bit semantic, but a lot of companies are very worried about these semantics, as is FDA. So where are we now? I think it's fair to say FDA is still feeling its way, although many companies are very interested in using genetic information in drug development and in getting that information into labeling. FDA has already issued several, quote, guidance documents. These are statements of agency policy that are in theory non-binding, but in practice sometimes have binding effect. Agency officials have announced recently they plan to issue several more guidance documents on a variety of topics: clinical pharmacogenomics, predictive and prognostic enrichment of trials, and how to coordinate internally. There are several centers under FDA, one dealing with drugs, one dealing with diagnostics, and one dealing with biologics. They all need to be talking internally so that if you are going to co-develop a drug with a test, that they're all on the same page about what the requirements are and their timing is coordinated, which is a big challenge. And they're also working on this relabeling issue, so the post-market problem for several drugs to include that genetic information. So I think it's fair to say it's very much a work in progress at FDA, a lot of interest in personalized medicine and using this new data, but a lot of challenges to actually getting there. And I will stop there. Thank you.
Well, thank you, Gail. And I think one of the most important things Gail told us was that the practice of medicine changed how it prescribed Erbitux and the other antibody against the EGF receptor a year before the FDA agreed to change its labeling. So medicine is paying attention to the evidence, and frankly, many of us think the FDA is very risk-averse. But there's a reason for that, and that's because the Congress that writes the budget for the FDA never congratulates them for success, but calls them in for panels in front of television cameras if a mistake is made. That's just a personal opinion. I'm going to introduce our third speaker, Roselle Kurzrock, by making one statement, because I see that at least half this audience is over the age of 50. When we talk about genetic, those of us that are over 50 remember that as inherited. And I want to emphasize that the gene abnormalities that cause cancer are primarily not inherited. The changes in the bases, in that 3 billion bases that make up the genome, this is not an inherited change, it's an acquired change in most cases. Damage that has occurred to the structure of the DNA because of things like the carcinogens in tobacco smoke, which cause 1/3 of all cancer deaths. So we're looking at a genetic disease, cancer, where the basis of the disease is not necessarily that you inherited gene abnormalities, but you can acquire them from sunlight, from tobacco, which is the main source, from carcinogens in the food we eat, and possibly from other things that we don't understand. That's just background. Now, Roselle Kurzrock is professor and chair of the Department of Investigational Cancer Therapeutics, and she's my colleague at MD Anderson. She received her MD at the University of Toronto. She runs our phase 1 clinical trial program, which does about half of our phase 1 trials, and it's the largest such program in the world. She has personally investigated over 80 new drugs in the clinic, has written over 330 peer-reviewed articles about her discoveries, and equally important, she created and directs the Masters of Science and PhD program in patient-based biological research at MD Anderson. So we give a PhD degree to people who are interested in studying human cancer. It's a very unusual program, and I'm especially proud of that program, and thank you, Roselle, for not only studying the new drugs on our patients, but teaching the next generation how to do it better. Welcome, Roselle.
Okay, well, thank you for inviting me, and what I'm going to talk to you is about what we're doing with personalized medicine in cancer. But to give you perspective, phase 1 trials, our first in human trials, are very new drugs. So what I'm going to show you is going to be about patients that are very end-stage disease, the most complicated patients possible. So patients don't come to us unless they've been told by their physician that there's nothing more that can be done to them. They failed all standard therapy. So that is the first perspective that I want to talk about. So to point out. Okay, so why do we need new approaches for treating cancer? Well, I think everybody in the audience knows intuitively that cancer, and especially metastatic cancer, is not a good disease. And when we look at the actual statistics, the mortality rate is going down, but it is a little bit slow, and at this rate it'll take 1,200 years to cure cancer. So we really need a new approach. And so why are things so slow? If we look at the classic way drugs are approved through randomized studies of unselected patients, these are the gold standard, and I've just listed a couple of standard drugs, a few standard drugs, but the data is similar for almost all our cancer drugs. The improvement in survival ranges from 11 days to 11 weeks. So the p-values are all significant, but the implications for the patients are much less significant. And the reason that the improvement is so small, we believe, based on the data, is that there's a subset of patients, about 15%, that approves a lot, and then the rest, the drugs are completely ineffective for. And we are not able to select that 15%, or we have not been able to in the past. We now have the technology to possibly be able to do this. So obviously this is a waste in life, and it's a huge cost as well. So therapeutic efficacy, can we improve? I'm going to give you some examples. Matching patients with targeted agents results in high response rates, even with brand new experimental drugs used in patients that have difficult to treat cancers. So we know lung cancer is a terrible disease and it's difficult to treat. But in a breakthrough discovery of this targeted agent that was then used just on patients that had the target, the small set of lung cancer patients that had the target, see what happens. Every part of this figure that is below the horizontal line represents a response. And as you can see, virtually all the patients responded. And this is a first-in-human drug, a brand new drug used to treat a very difficult disease. And instead of having a small response rate, you have almost every patient respond. So what do we know about this particular abnormality? We know it occurs only in 4% of lung cancers, and it also occurs in 2% of breast cancers and 2% of colorectals. And this illustrates the entire problem with the molecular phenotyping or genotyping. Each subset is going to be a small subset of patients. But if we can pick out that subset, we can get very high response rates. And the data has now increased. We know that common tumors are very heterogeneous, and small subsets are important. And in fact, the reason that tumors are common, the reason that lung cancer, breast cancer, colorectal cancer is common, is almost certainly because there's many different pathways to get there, and therefore there are many different subsets of patients. So the classic way of doing things and developing drugs, of doing randomized trials in unselected patients, mostly gets you the wrong answer. It in fact tells you that the better drug is actually the drug that hit the most common target. And since all of those targets are seen in only a small percentage of patients, that's why we get such minimal improvement. So what else do we know about targeted therapy? Well, I think most people that have known people that have gone through chemotherapy, a lot of patients wonder, is the drug worse or the disease worse? But many targeted agents, actually, because they've been developed specifically to the target in the tumor, have very little in the way of side effects. So the side effect profile is much better. And I'll just give you a few examples. This is a young woman with a disease called Castleman's, and we know the target is IL-6, and these are her skin tumors. When we gave her a drug to target IL-6, she actually said that she could see the tumors disappearing within 2 hours, and she's now been in remission for 3 years. But she jogs 5 miles on the day that she gets treated. And this is not, I'm not going to tell you all targeted therapy is that easy, but many of them have a much better side effect profile, or even no side effects in some cases. The other thing that we know is that even patients with advanced end-stage disease can respond if we know the target. So this is a patient also with Castleman's, and this man, young man in his 30s, was on dialysis because his kidneys had failed, and he'd been on the ventilator 3 times, and in fact was on the ventilator when we started treating him. And now he is doing great, is in complete remission. And we know that almost 80% of these patients, like with the lung cancer patients, because we've matched the drug and the target, can respond to this drug. So while it is often claimed that we need new drugs to treat cancer, and that's almost certainly true, but there's a more fundamental problem, and that may be in the way we classify cancer. And it's very well understood that, for instance, insulin is a great drug, but not if you use it to treat pneumonia. And similarly, pneumonia would be a very difficult illness to treat if we used insulin rather than penicillin. So in the same way, we may have excellent drugs, but they work poorly because we treat patients with the wrong cancers with them. And that's what we've been doing for the last 30, 40 years. But we now have the technology now to be able to begin to change that. So we've been involved in something that I'm calling the Cancer Reclassification Project. And current cancer classification uses the light microscope, an organ of tumor origin. And most of you know light microscope was invented in the 1600s. And so, but that's not enough anymore. I believe that the cancer needs to be reclassified using, quote, the molecular microscope, which is the genetic screening. And this needs to be added to augment the light microscopic diagnosis. And then patients need to be treated with drugs targeted specifically according to this classification. So this is one last project I want to show you. These are 2 major signaling pathways in cancer. It's called the PI3 kinase pathway, which is on the right. And we now know that there are mutations that occur in each part of that pathway, and we also have drugs to target that pathway. And there's also a resistance pathway called the RAS-RAF-MEK pathway, and we have drugs to target that. And we even have combinations of drugs which target both pathways. So what happens if we look at, for instance, PI3 kinase as an example, the first thing, we've looked at over 500 patients, and what we found was 11% of patients with advanced cancer have mutations in this pathway. And what you'll notice is this crosses disease boundaries. So it's in endometrial cancer, it's in breast cancer, it's in bowel cancer, ovarian cancers. So all of these, what are supposedly different types of cancers, now have something in common, and that is a mutation. And this is what I think we need to be targeting, not just looking at breast cancer as breast cancer, but what is the molecular aberration in it? And then if we look at what happens when we look at several of those molecular abnormalities that I just showed you, even end-stage patients, if we treat them, it doesn't matter what drug we're using, and it doesn't actually matter what mutation. Here the concept is just matching. Just by treating them under a matching program, we have a substantial number of patients that show a response. Again, the response being any patient where the graph falls below the horizontal line. And furthermore, if we look at their survival, this is not a randomized study, so it's not your gold standard, but I still want to show it. If we treat patients by matching them with the drug, you get the red line. 75% of these patients with end-stage disease are alive at 18 months. If you treat them the way we used to treat them, which is we look at the diagnosis by the light microscope and put them on the targeted drug, 50% are dead by 6 months. So we're already, even in end-stage patients, being able to see real differences in survival. So what is the difference? So treating patients with molecular matching, we get partial and complete remission rates of 30%. Treating them the old way, 4%. And I mentioned the improvement in survival, 75% alive at 18 months versus half the patients dead by 5.8 months. So what I want to finish off by is saying, so you might say, well, 30% isn't such a great response rate, and that's true. But keep in mind what I said in the beginning. I'm treating patients who are at the end, who failed all therapy. And there's a story that I think is worth paying attention to, and that's the chronic myelogenous leukemia story, which was probably the first big successful use of targeted treatment. And when chronic myelogenous leukemia patients were treated with a matched targeted agent when they were end-stage disease, the response rate was 15%. But when we treated newly diagnosed patients, the response rate became over 90%, and the median survival increased from 3 years to 25 years. Slides don't want to move. Okay, so I want to finish off by saying that I've been concentrating on the tumor, but I think the host is also important, and that I think the host will be more difficult. I think the tumor is the first step, but probably this lady who is obviously a chronic smoker and lived to be 100, this was probably not good luck. This was probably genetics. And so the near future, diagnosis of cancer should require advanced molecular technology for every patient. This is my opinion. Match patients with drugs based on the molecular profile. Treat newly diagnosed patients in this way rather than just those with end-stage disease. And rethink approval of drugs based on large randomized studies of unselected patients resulting in significant p-values but clinically insignificant benefit. Thank you.
Well, I jotted down some questions for our speakers, but I'd rather start out by asking the audience. You've been very patient and you've heard an awful lot of information. Who would like to raise some questions? And I think, are there microphones for microphone here, so we'd be glad to hear from you. Okay, well, maybe I'll kick it off then. It's pretty expensive still to sequence a genome, but we do have patients coming into MD Anderson carrying with them their genome sequence. You can buy it today. You can go have it done. So I've got this kind of cancer, doctor, and here's my genome sequence, and what are you going to do for me? And of course, that brings up how do you educate doctors to handle that information. But let's ask the question to the panel, is it now worthwhile for everyone in this room to go out and have their genome sequenced and spend the 10 or 20 or so $1,000 to have that done?
I'll start. I don't think yet. I particularly don't, I'm not particularly comfortable with the scenario that you just presented, which is that individuals going off and getting, whether it's a genome sequence or whether it's some other type of genetic test that they can get without a physician being involved, and then in some ways ambushing their physician, many cases who are really not prepared for that information, and actually some of that information is probably not even, we're not even at a stage to fully understand what it means. But with that said, I'm much more comfortable with the idea of, and I think you heard it in particular from the last speaker, that increasingly physicians are going to come up against circumstances. And cancer will be some of the leading ones, but there's other examples as well where this is now standard of care to either get the genome sequence or a tumor's genome sequence or some set of genetic tests. And I think it's with the health professionals in the middle of that is the thing I'm much more comfortable. So it is a wild, wild west out there, and all you have to do is go to the internet and go to Google and type genetic testing, and what you get back, you'll be shocked to see. And it is a free market, and you can do lots of things, but I'm much more comfortable seeing this done within the context of a healthcare delivery system and a physician or healthcare provider-patient interaction.
Good. Anyone else? Let me just say again, we're talking about sequencing a person's genome in order to predict their risk of having a disease. That's different from taking the tumor that they have and sequencing the tumor to see what's wrong with that particular tumor. And again, many of the gene abnormalities in that tumor were not inherited. If you studied the rest of their body, you wouldn't find those problems. So please distinguish between those two. Ambassador Jurigian.
Right. The non-scientist in the group, but I'd like to ask the panel, what is the nexus between nanotechnology and matching patients with drugs using their molecular profile? Is this the hope for targeting medicine to specific cancers?
I think nanotechnology may improve some of the targeting, but many of the drugs that we're using are not nanotechnology based. So I think it's another methodology that can improve target, but certainly not the only one.
Yes, Neil. John, go ahead.
Yeah. The discussion is focused on genomics. Wondering, especially as we get, Eric, to the later stages, you were talking about what you see as the role of proteomics, metabolomics, et cetera.
Sure. So the genomics is the comprehensive study of the genome, but of course the genome is basically the software to produce all the building blocks of cells and tissues and organisms, and those are proteins is the one that comes to mind immediately, and then those proteins interact and yield products of different chemical processes or metabolites, and what we're just talking about here specifically relates to genomics, but there are many developments that are still taking place with technological advances are giving us the ability to look at tissues or look at body fluids and get a survey of all the proteins that are there and all the different metabolites that are there and all the different, and they're very different technologies. I don't think they've accelerated as fast as those for sequencing DNA, but absolutely, that's all part of the future. And even the question about nanotechnology, there certainly are technological advances, I think, that will be even changing the way we do diagnostics of non-DNA components such as proteins and metabolites. It's just not particularly my area of expertise, but they're absolutely in development.
Back in the back.
I'd like to ask a question that goes to fiscal policy, if you will. The Bush administration underfunded NIH for several years for whatever reasons.
Which Bush? We should be clear here.
W.
Okay.
Because of the wars,
Yeah.
whatever, you know, the tech wreck, et cetera. Now we find ourselves with the Obama administration having increased funding to the NIH through the stimulus program. That funding is likely to start deteriorating over the next couple of years. And what we have is sort of an intersection of opportunity, again, running up against, let's say, limited funding or risks of funding, at the same time that we're seeing pharmaceutical companies and biotech companies being pressured on drug prices, so they're not going to have money to reinvest. But we have this great opportunity ahead of us. Pardon my being long-winded, but I have a son here at Rice, and a couple of years ago when he was interviewing colleges, I kept asking admissions people, where are the predominance of kids going? Where's the real change in students entering school? Where are they going? And they were all going into the life sciences. And I thought, this is a great opportunity. But is this lack of funding or the risk of funding going to squander our opportunity in this area and maybe cede it to China or other countries?
I suspect you're going to make me take this one, aren't you guys? Mm-hmm. Yeah, I had a feeling. I resonate completely with what you say. It's very frustrating, and especially the position I have now, where the opportunities are absolutely overwhelmingly positive, and all you want to do is push the accelerator. Because you can see, and all you have to do is visit cancer centers in particular, and you just see, because I really do believe, I'm not just saying this because who our MC is, I really do believe that cancer genomics is going to have some of this highest impact on patient care of all the genomic developments over the next 5 or 10 years. And it's just at the time when we just want to push that accelerator as hard as could be, because we truly see that this is going to yield positive developments. And yet we spend so much of our time just trying to figure out how to deal with what could be a pretty significant problem in budgets for the next couple of years. So it's very frustrating, is what I can tell you. I mean, it is a little bit of a white knuckle experience going on at the NIH right now, of not knowing exactly what's going to happen. We're coming out of this $10 billion stimulus situation, which was catnip in some ways, but it does not give the kind of sustained funding that's absolutely essential. You know, we've just got to hope for the best, but it's what keeps me up at night. And it takes a disproportionate amount of scientific leaders' time, instead of really thinking about the best applications, of just worrying about how to sustain any sort of momentum. And by the way, the last thing, I don't mean to be wrong with, I worry about your son. That's actually what I worry about as much as anything else, because I worry that he'll get discouraged if he sees that the life of a physician scientist or of a basic scientist is one of cycles of funding that just don't lead to a productive career. I worry that he'll decide to do something else. And so it's the next generation I worry about as much as anything else.
On that nice note, Neil?
Neil, you're going to fix this for us, right?
Let me say a nice word about the NIH. Is this on?
No, but if you're going to say a nice word, turn the volume up, please. So I wanted to stop.
NIH often gets criticized for not managing its budget better. But what Dr. Green, I think, probably won't want to say is that if Congress comes and says, we'll double your budget over 5 years and spend it wisely and spend it quickly and get it out the door before the next appropriation cycle, and now we're going to cut you off at the knees, how come you didn't manage that very well? Well, I think this audience understands that. So I think they do a great job, but boy, there are tough times coming, I think. I want to ask Dr. Green a question about his slides. You indicated that one of the big barriers to progress is now in the computational bioinformatics side. In the strategic plan, the diagrams you showed us, when you're projecting progress, is your assumption about computing Moore's law, or what if there were a major revolutionary breakthrough, nanotechnology or paradigm change or architecture, anything else
in computing, what impact would that have on your future?
Yeah, I mean, we don't have the solutions to it. In fact, I believe the computational challenges that now face biomedicine are bigger than just the field of genomics. But we are hopeful that there will be some advances. And in particular, what we've seen happening over the last even 3 or 4 years is, even if you go back to the Genome Project, go back 10 years or something, the truth was computer scientists, real strong academic and even private sector computer scientists, weren't particularly interested in genomics. They said, you know what, your data sets aren't that impressive. We want impressive data sets. We do planetary science. We do particle physics. Those are big data sets. That's changed in the last 3 or 4 years. Now, all of a sudden, computer scientists, great minds in computer science, are gravitating to genomics, say, oh my gosh, you finally have the kinds of challenges we want. So there's ideas that are bubbling out there, whether it's cloud computing, is that the solution for our universities, whether it's new technological advances that are going to reduce the cost. I think all of those things are going to be needed, but I really want to stress the third component of that bottleneck I talked about, and that's workforce. Is that even if we solve this all with hardware, what's very clear is we need people who are cross-trained, who really can speak the language, can speak to the physician scientists, at the same time know how to handle big data to serve as that, but really know biostatistics and the kinds of algorithms they're going to need. Those individuals are going to be key parts of these teams of the future. And maybe I never said it in my talk, but moving towards genomic medicine is not going to be individual investigators marching this along. This is more and more team-oriented, multidisciplinary science. And we need all of those players of their appropriate expertise to come to those teams.
I'd like to agree with that. And for the gentleman whose son is at Rice, anyone going into biomedical research who also really understands computer technology is going to have a much stronger chance to get a good academic appointment 10 years from now. I would guess half the time spent is going to be in front of a screen and analyzing data rather than doing experiments. Now, when I started, 95% of your time was doing experiments.
Completely agree. Completely agree.
Judy. You got a loud voice, Judy.
A little help here. This is really a layperson's question, but recently a number of us in this room were offered the opportunity to be in a gene study at the University of Texas
Health Science Center.
Well, Seraphim Molecular Energy or whatever, molecular biology, with Baylor. And the study is to study schizophrenia, which, you know, usually occurs in a young person, so I think some of us are past that age. But I jumped on this because they said that we're going to take your blood and we will analyze it for 200 to 300 genes directed at that particular study. And I said, well, but if you see something else, I certainly hope you'll call me. What kind of things else might come out of this for each of us individually? I mean, would they be able to call and say, you have a certain type of gene for breast cancer or ovarian cancer?
You want to take it?
I'll take a crack at it. So what you're referring to is a very big debate among scientists and also ethicists about returning results. And the traditional view, if I can try to characterize it, is we don't return results because this is not clinical information, it's research, and we don't want to tell you something prematurely. From some of the social science research that's been done by the Genetics and Public Policy Center and others, patients or people who participate in these studies often do want to know. So there's a bit of a disconnect between saying, I understand it may not mean anything to me, I'm not taking it as gospel or clinical information, but I want to know. And so this debate is going on. But it's the people who design the study who ultimately are going to say, yes, we will share the results, no, we won't, or somewhere in between.
Judy, I hate to, maybe Eric would disagree with this, but most of the genes that are detectable today in this kind of a screen do not have that big an impact on health. The list of genes where you'd say, please give me a call, something like BRCA1 for breast cancer, that list is very short. It's 24 genes or less. So you're not losing a whole lot of information today. I hope in 10 years, your question, the public will have said, look, there's 1,000 or 2,000 genes that might affect my health. give me that information if I donate my blood. What do you think about that, Eric?
Well, I think your question is just a small glimpse into a future that's going to be much more complicated, and your question will even be more relevant even a year or two. In fact, I wouldn't be surprised. For example, I will tell you at NIH, we've had going for several years now a project where we're taking 1,000 people from the Bethesda area, and we're sequencing their whole genome in a study very similar to what you're describing. The initial focus is on cardiovascular disease, but we actually, by sequencing their whole genome, we're looking at all 20,000 of their genes, and we're seeing all sorts, and there's many examples we now have where we have found things where we have gone back to the individuals. They gave us permission to come back to them and say, we have some things that we now know about you, and in some cases they were at greater risk for several diseases, and we were able to intervene and actually help them. But I think this idea of 400 genes, 200 genes, it's going to be the whole kit and caboodle in a matter of months, because it's going to be just as cheap to do that by generating. And there'll be lots of people who will be participating in these studies. And actually, that's a good thing. We need large numbers of people to really dissect some of these complicated genetic diseases.
And if we all do it, there's not a computer in Texas that could store the data. I mean it. There's not. Yes.
The last speaker pointed out genetic problems that were recognized that were cross-cancer types, 20% breast cancer, 20% lung, whatever. And yet I know that traditionally your departments are organized into leukemia, breast cancer, what have you. Do you see this leading to a reorganization and a restructuring of the way you practice medicine at MD Anderson in that regard? I know that my wife has had a form of leukemia, and we have noted that the classifications have changed, with the World Health Organization and others have taken it out of that slot and put it in this slot, and now I understand with greater understanding of the genome and what's behind it all, they're going to be able to reclassify it again. And I just wonder, when you go to a leukemia department, are they going to now be more sophisticated in the sense of looking at your particular type and causation of your leukemia or your breast cancer? How is that going to affect your organization?
Well, I'll try to answer that question. The answer to your question is that leukemia is very different from breast cancer, and we're still going to need doctors that specialize in leukemia and doctors that specialize in breast cancer. But we are setting up at MD Anderson, and the man doing it is sitting here, Dr. Stanley Hamilton, the capability of interrogating leukemia tumors or breast tumors or colon tumors or lung tumors to see which genes are abnormal in those tumors, and then assigning therapy based on what's found. And I, not to make it, put a downer on it, but it's much more complicated than just what the tumor genome sequence is, because the person that has the cancer may be feeding the tumor. If the tumor gets into some people's bones, it may grow, and if the tumor gets into other people's bones, the same gene problem, it might not grow because the bones are secreting different chemicals to stimulate the tumor. So it's a very complex thing, and it's still important for leukemia specialists to know everything about leukemia and breast specialists to know everything about breast cancer. That's my opinion. Now, Rozelle might have a different opinion. I don't know.
I think that there's going to be two stratifications, one by the classic light microscopic diagnosis, which is breast cancer, colon cancer, and the second stratification will be by mutation. I think what's not totally clear at this point is which will be the primary stratification, whether the primary stratification will continue to be organ-based, or whether there'll be some drift to primary stratification by mutation with substratification by organ. I still think the organ is going to be important. I don't think the light microscope is going away, but if you have breast cancer, you're going to need a molecular subset stratification. That's the bottom line, or vice versa.
Although it's interesting, your question touched on an issue, and we only just talked about it through the lens of cancer. And the other thing happening in genomics now, if you move away from cancer, is sort of where in the medical profession should sort of the expertise reside in terms of understanding not cancer genome sequences, but just individuals, your germline, your own DNA, not non-cancerous DNA, those sequences. I will tell you, I'm trained as a pathologist, for example, and I will tell you there's a very critical look going on now in the pathology community of should they be completely retooling to become the consultative experts about individuals' genome sequences. Or meanwhile, at our institute, we run a medical genetics training program for medical geneticists to be trained. Should they become the experts, or should every medical discipline, whether you're a pediatrician or an internist or an obstetrician, they're all going to need to learn a little, or actually learn a lot about genomics. The question is, should it be spread across, or should certain disciplines really step up and become the genomic experts? I think cancer is almost a subspecialty of that. And I don't know where this is going. And if we keep marching on, we could start thinking about, practicing physicians are not ready for what's happening. And even the current medical curriculum as it exists today is completely inadequate for what's happening in genomics, not to mention the general public getting their literacy up higher. So there's a lot of issues around all of this. And the faster things move, the harder it is for everything else to catch up.
So doctors have to get retrained all the time. Yes, in the back, there's a question.
Question related to Roselle. I think I was
closer.
I was interested in her comments about the efficacy that's been found in our historical treatments at a low 4%, and now our ability to be more successful. What's your intuition on the impact on the pharmaceutical industry under the fiscal pressures everybody has? Are they going to slow down because they don't think they're going to target enough people to use their drugs? Or are we going to see the same drug being used across a wider variety? That backs to the FDA issue, but therefore continue the pace or accelerate the pace of therapy development?
So just to be sure, so the 4% that I mentioned pertained to our population, which is end stage. It's probably more like 15%, which is still not great when you treat patients earlier in the classic way. Not long ago, drug companies didn't want to go after rare subsets of disease, but I think that changed actually with the chronic myelogenous leukemia story, because Novartis is making a fortune on that rare disease. And so I think that other companies are looking at these subsets as being more viable. In addition, I think what one has to remember is that it may be 2% or 4% of lung cancer, but that's still a lot of patients. And if you add in a percentage of breast cancer and a percentage of colon cancer and a percentage of lymphoma, it's still less than what they would have gotten before, but it's not insubstantial. The difference is now that you're actually treating patients effectively rather than treating a lot of patients ineffectively. So I'm not sure that, I don't actually think it's going to dampen the enthusiasm.
I would agree with that. I think, you have to remember that most of the new drugs that enter the clinic against cancer fail today. And the cost of developing a successful anti-cancer drug is estimated to be $1 billion, and it takes 10 or 15 years. And with the kind of data that Roselle showed you, we can hope that it'll take 5 years or less to develop a new cancer drug, because you'll be treating the right patients that are likely to respond, and it won't be a 3% response rate, and it's tossed aside and wasting a drug that might have worked, and we can get right to an FDA registration trial within a year or two. Isn't that right, Roselle?
Yeah, I think that's a good point. In fact, one of the drugs that I showed, the anti-IL-6, went directly from phase 1 testing into a phase 3 registration trial without any intervening phase 2. And we've had other drugs like that in our program as well. And I think conceivably, and crizotinib, the lung cancer drug, exactly the same thing. So this can be a way, if utilized properly, to shorten the development period.
Which I hope will reduce the cost. That's up to the company. Other questions? Yes.
I'm hearing sort of a sequential thinking. Let's look at the sequence in an individual patient, find out what it is, normal, abnormal, inherited, acquired. What does it do? What does it produce? And attack the products. Now, what can be done, or is being done in research, to try to go to the root cause and change the sequence itself?
Change the sequence itself in the individual. So realistically, being able to do that to your germline is probably fraught with all sorts of technical problems and probably is not going to be a realistic way of changing the course of someone's progression and disease. Now, with that said, there's great promise for some forms of gene therapy, but where you have success in gene therapy is not re-engineering what exists there, but augmenting something that's broken. So in the classic cases of gene therapy, it involves diseases whereby there's a protein that's not being made because the gene is broken. So you remove some bone marrow, typically some bone marrow cells or blood cells from an individual, and you add back a good gene as an extra copy. You're not stitching or fixing what's already broken. You're just augmenting, and then a good protein can be made. You put it back in the bone marrow because bone marrow is a good place to put things and grow. And now that's great for diseases of the blood in particular because we can access the blood through the bone marrow. That doesn't work for many, many diseases. For example, those that affect the brain or those that affect tissues that it's hard to introduce genes. But there are some successes in gene therapy, but not in the way of actually fixing something. It's more by adding a gene that's broken. Now, there are some promising developments in cancer. I don't know if you want to comment on any of those where you're engineering.
I think cancer's in the same boat.
Yeah.
I think attacking the product of the gene is going to do much better than replacing a broken gene, although there's been, 20 years of research going on in that, and so far it's been a failure. And it's because when you try to replace a gene, you have to get it into the cell in ways that there's risk to damage the cells. The gene is packaged in a virus that could do damage to the cell in addition to replacing the abnormal gene. You'd think the car brakes aren't working, fix the brakes and go right to the source. And we'd love to do that. I think it's going to take a long time. I agree with Eric. I think we have time for maybe one or two more questions, and then people have been very patient. It's
one over there.
Good. I can't see it through the spotlight.
What are the implications for this sort of technology on the insurance industry as far as coverage goes and using this information against you?
Why are you looking at me?
Oh, you're going to eat this one up.
It's a great question. So the issue of what insurers as well as employers can and cannot do with genetic information that may predict risk of disease has been a huge debate that was only recently resolved. It took about 12 years for Congress to enact what's called the Genetic Information Non-Discrimination Act, which they did in 2008, which says basically that health insurers and employers can't require you to take a genetic test as a condition of insuring and or employing you. And also, with limited exceptions, can't even request that you do that. And part of the concern, I mean, there's been a huge literature, huge public concern about that very question. Well, if I'm going to know this, who else is going to know, and what are they going to do that's adverse to me? Part of the concern in the research community was people wouldn't enroll in clinical studies because, for the example that we heard before, well, they're going to find out something about me and it's going to have a negative impact on me. I don't know in practice whether the passage of GINA has resulted in people being more willing to participate. I'd be interested to hear more about that. So the short answer is GINA, but I said health insurers and employers. There's lots of other people who might want to know your genetic information, your next door neighbor, your nosy in-laws, and the law really hasn't caught up there in terms of people's ability to test you without your knowledge. So I think that's the next frontier on genetic privacy.
Well, I'll give a dissenting opinion. I think if you asked anyone, give me a list of the examples where people have abused genetic information it would be very short. Maybe you have more information
no,
than I do.
I think that is...
So we're assuming let's get rid of all risk. And if we assume let's get rid of all risk, we're not going to get anywhere on this planet. People are going to cheat. People are going to use information improperly, but so far, in spite of all the genetic information out, nobody has been hurt by it so far. And if we design a system to protect against the cheater, we're going to destroy our chances of benefiting from all this wonderful information. So I'm a risk taker, and then punish the people that cheat and put them in jail for 10 years so nobody else will cheat. If we design it in a way that totally protects all this information, it's going to really slow down what happens, in my opinion. But I happen to look at it from the point of view of advancing science and helping people as quickly as possible. The other answer to your question is the insurance companies will pay for this because we will demand it. As the public gets real information that this affects cancer outcomes, we're not going to sign up for a policy unless it covers that. And the best example of that is colonoscopy. I'm sure everyone in this room knows that if we all get colonoscopy at age 50, and then every 5 or every 10 years after that, we can cut the death rate from colon cancer 50%. Now that's the second most common cause of death from cancer after lung cancer. And at first, insurance policies would not pay for a colonoscopy unless you had symptoms. Well, the whole point is to get it when you're not symptomatic and find polyps when they're not causing any trouble and getting them out. And when the information came out that the colonoscopy could cut death rates by 50%, the American public demanded that when they signed. Every July you sign up for your insurance policy, and if it didn't cover colonoscopy, nobody signed up. And within a year or two, all the policies covered a colonoscopy on a healthy person, which costs the insurance companies around $1,200. But we'll demand it, and I think it'll happen. Will it make medical care more expensive? I don't think so, because these are very expensive drugs. And if Rizal's numbers say that only 15% respond to a drug that costs $10,000 a month, the insurance company should be very happy that the other 85% aren't getting that drug, and we're saving that money. And the economics of that hasn't been worked out in detail yet, but my suspicion is that actually it'll save money, even though we're spending money up front to do these tests. I don't know your time situation. I'm only 20 minutes over time.
People aren't leaving.
All right. Is there one last burning question, or should we call it a day together? There's a last burning question.
Burn, baby.
This is a layman's question. Let's say you live in Tyler, Texas, and you go in and it turns out your doctor tells you have colon cancer, and you go to the local cancer doctor. And from what I've heard here, it seems likely that if you have colon cancer, it would be a good idea to have a molecular analysis. But are you going to get one in Tyler, Texas? How does that patient, let's say it's me, and I know that it would be an excellent idea if I got that analysis, how do I get it? It's fine if you go to MD Anderson for your care. You're probably, that's not a problem. But how about Tyler, Texas?
They'll be able to get it in Tyler, won't they, Rizal?
I think that there's an increasing number of commercial groups that are offering molecular diagnostics. So you're going to get more sophisticated testing at MD Anderson, and that's not a surprise. But for instance, the RAS mutation that was mentioned by Dr. Green, you're going to get that very likely going to get that in Tyler, Texas as well. Probably at a tertiary care center, you're going to get more, but it is drifting down into the community, and there are commercial agencies that are providing for that type of testing.
And if you can't get it in Tyler, maybe you can go on the internet and get it.
It's not an emergency test. It's not an emergency test. You can wait a week. And the biggest testing institution in the world is in Beijing. And people from the U.S. today are sending stuff to China. It's tested. You get the results back in a week. And I believe that maybe in Omaha, right in the middle of the country, there may be a huge genetic testing center that can do it cheaper than any individual hospital. That's all to be worked out in our wonderful capitalist system. But I think you'll be able to get your test done. As it's proven to be efficacious, hospitals will have that test available on site or farming it out. Well, it's been great questions. We've had a wonderful group of speakers, and thank you for attending with us, and thanks to our speakers.
Sure. Likewise. Good to see you. Thank you. Hello, Eric. I'm Bob Wells. Hi.